Well, Bach did an outstanding job of kind of covering so many bases in renal cell carcinoma. And I'm today just going to talk about one type of RCC and share some findings that we have about not only the metabolism in the cells, but also importantly, the ultrastructure in the cells. So, and I will not belabor this slide other than to say that I think kidney is a very interesting organ. Human kidney is an interesting organ in so many ways. It does so many functions. It pumps so many different ions. It regulates salt. It regulates red cell production, all these different cells that have different functions. And so I think that is part of the reason why we get such a variety of different histologies in renal cell cancer in humans. And so I think it's a cornucopia of different metabolic phenotypes, different genes that are causative, and different clinical courses and aggressiveness. And so today I'm just going to focus on this one here, FH deficient renal cell carcinoma. That's what we study. And in these patients, it's a hereditary syndrome. It's inherited on chromosome 1 in an autosomal dominant manner, which I'll go into in a moment. The patients can get, but not all of them get, these skin leiomyomas, which are basically the erector pili muscle in the skin overgrows, and they can be painful, but not carcinogenic per se. Women in these families are at risk for development of uterine fibroids. Again, that's a proliferation of smooth muscle, and it's associated with the syndrome. And a subset of these patients, not all of them, but a subset will go on in the course of their life to develop one or more renal tumors of this very special histology that I mentioned, which is formerly known as type 2 papillary, but the WHO has decided that that is not the right term for it anymore. So now it's known as FH deficient RCC. These tumors are extremely glycolytic. This is a PET scan from a patient with a tumor, an FDG PET showing glucose uptake. The tumor also accumulates fumarate, and that's because it's lost the function of this TCA cycle enzyme. This is the TCA cycle cartoon I have here. And the enzyme FH interconverts fumarate and malate. And so the consequence of losing functional FH in these patients is an overaccumulation of fumarate. This remodels metabolism, increases pentophosphate pathway activity, causes the cells to use glutamine differently than most cells do. This is just an example of a pedigree of a family. Two families actually, for a mutation that we discovered in their FH gene, just showing the manifestation so that you can have cutaneous leiomyomas, you can have uterine leiomyomas, and then, as I mentioned, you can have these RCCs. And so this particular family here had both types of leiomyoma and are very worried about whether they were at risk of developing RCC. And so we followed them up and found out that they had an intronic mutation that was not detected by normal sequencing of the FH gene. After we found that information, we found another patient and family who was affected with the same mutation. So the general idea here to show this slide is that it's autosomal dominant inheritance pattern. So you only need to inherit one affected copy of this gene to be at risk for these phenotypes. So what happens in the tumor, in the affected cells? So the one thing I think is most interesting about this and so many other hereditary cancer syndromes is the tissue specificity. So these people are losing, they stochastically losing a copy of chromosome 1, or at least where the FH gene resides. And bizarrely, all they end up with that we know of are these benign skin lesions, benign uterine lesions, and then this super aggressive form of kidney cancer that's needs to be treated as aggressively as possible. So the way this works is with a wild type allele and an HLRCC allele, an FH allele, mutant allele. We can have two things happen. There's actually more than two, but the two main things that can happen is we could either have a point mutation in the residual wild type allele, resulting in compound heterozygous mutations in the FH gene. Or what turns out to be most common is loss of heterozygosity. So the entire chromosome 1q or a region of chromosome 1q breaks off, and the result is that the cell is left with only an inactive copy of FH. So as I mentioned, that results inside of the tumor of this massive accumulation of fumarate. And this ends up being a tool that we can use to characterize this tumor pathologically. On the left is renal cortex tissue with inflammatory cells and some other funny looking cells down here on the left, H&E stained. And a researcher by the name of Norma Frizzell, studying obesity of all things, got interested in this modification that can happen to proteins where the cysteine thiol interacts with fumarate, which is an electrophile. Fumarate can actually covalently bind to cysteine protein thiols, protein cysteine thiols. And so that post-translational modification is non-enzymatic and, to our knowledge, non-reversible. And so cells that have a lot of fumarate in them accumulate this mutation, and we can stain immunohistochemically for the abundance of this 2SC PTM. And you can see in this section that it's just lighting up the neoplastic cells in a sea of other cells, immune cells and tubules and connective tissue and vessels. And so it's a very useful tool here. And I'll come back to that in a moment. Dr. Maria Marino in the laboratory of pathology has used this extensively and characterized this as a diagnostic tool for clinical diagnosis in our patients. I'm a biochemist by training, kind of a jack of all trades, but a biochemist by training. And my big question with this tumor type is what's causing this Warburg effect in the HLRCC tumor. So I showed you that they take up a lot of glucose. They also show a propensity to ferment lactate. This was a question that Warburg was asking over 100 years ago, because he found that tumors tended to ferment lactate even if oxygen was present in the experiment. And so our approach to studying this is to use very fresh tissue from the operating room. We really aren't studying mouse models of this. We do have preclinical models, xenograft models, but mainly my interest is in looking in the human model. And so we try to procure these tissues as quickly as possible because metabolites change quickly. But it turns out that I think anyone who does EM knows that organelles can also change during ischemia. So, a tissue that comes out and then sits in the OR waiting to go to the frozen room to be processed by pathology for an hour or two, or even worse, sits in the body without a blood supply until the end of the surgery because it's a robotic surgery, the mitochondria are not going to look the way they did when the thing was perfused well with blood and oxygen. Here's just a few examples, and I'll be quick, of patients that we study. Dr. Linehan has been studying these patients for over 40 years. And we've had a number of them come through with primary tumors or locally advanced or even widely advanced disease. This is just showing that this left renal tumor is hot on FDG PET. It has this very papillary, beautiful papillary structure where you can see kind of a basolateral and an apical orientation of the cells. And what we do is we derive cell lines when we can from these tumors to try and study them further molecularly. Here's an example of another cell line that we derived. Slightly different histology, but still papillary looking. And if you notice that there's this very angry nucleus phenotype in this type of tumor, large nucleus, large nucleolus, and sometimes you can find some clearing around the nucleus, which Dr. Marino calls orangiophilic nucleus. Here's yet another example of a patient where we derived a cell line. Again, large nuclei, angry nucleolus, and people have said that they look like they're virally infected nuclei. I think that might be an interesting thing to think about further. This is just the sentinel PET scan from this patient who unfortunately had widely metastatic disease up into the mediastinum and into the ribs. So then we're talking about EM today, and so I wanted to show you what EMs look like, TEMs look like from tumors that we obtained from these patients. So on the right is a normal kidney region from a nephrectomy from one of these patients, so that the patient is harboring the FH mutation, but they have no known problems with their kidney function. We see mitochondria with really nicely packed cristae, abundant mitochondria, nice double membrane. Multiple cells are in this plane of view. Going over to the left, in a primary tumor, the staining isn't so great, but what you can see is mitochondrial structures, but the cristae are really malformed or absent. There's a little bit of an onion-shaped structure forming in some of these mitochondria. And if you look in regions like this could be artifact of fixation or staining, or it could really be that the membrane in this mitochondrion is rupturing or losing integrity. Here is a metastasis from a different patient. Again, fewer cristae, enlarged mitochondria, and some of these sites like here where it's not clear, but perhaps there's breach sites where mitochondrial contents could leak out. A very smart colleague of ours in Europe studied a mouse model of this disease and observed the departure of mitochondrial DNA from the mitochondria into the cytoplasm, which was inducing the C-gas sting response. And we're curious to follow that up in the human model. But one thing I'll point out is that it's not that these cells don't have mitochondria. The mitochondria are there. So we started to characterize them. These are some mitochondria from the cells, cell lines that we have, which I won't go into too much detail, but you can see, again, disrupted structures. Here we have cristae that are very, very malformed, a lot of open space and swelling. I really don't even know what's going on with the cristae in this cell line. But in general, problems with the mitochondria, but they are not absent. So when we look in the tumor biochemically to look at the structure and function of the respiratory chain, so that's the part of the mitochondrion that's reoxidizing NADH generated in the Krebs cycle, and it's also converting succinate to fumarate in complex 2. And all of these electrons are traveling through ubiquinone and then eventually through cytochrome C to complex 4, which is cracking molecular oxygen that we breathe into water. So just looking at the assembly of complex 1, this is a native gel activity assay. And so up here, the blue bands mean that complex 1 is functional in the mitochondria of these two normal kidney mitochondrial preparations. Whereas in these two HLRCC FH deficient tumors, complex 1 enzyme activity is greatly decreased. That decrease in activity is associated with a profound decrease in the assembly of complex 1. So these tumor mitochondria are unable to assemble complex 1. And we showed that this is also the case for a number of the other respiratory chain complexes and supercomplexes. We started asking why this is happening. One assay to do was to count mitochondrial DNA copy number. We find that compared to renal cortex samples, although there's still mitochondrial structures in our primary and metastatic tumors, mitochondrial DNA copy number is greatly decreased. The mitochondrial genome encodes subunits of complex 1, 3, 4, and 5 in the mitochondrion. So a diminished amount of these proteins being able to be synthesized may explain, at least in part, the diminished assembly of complex 1. However, at the same time, we went and sequenced a whole number of tumors and tumor cell lines, and we found that the mitochondrial DNA was really peppered with these different point mutations and frameshift mutations. And in the case that we saw mutations in the cell lines, we typically saw those mutations in the tumors at varying heteroplasmy, which means that these things were arising before we put them into culture, that the tumor in the patient was accumulating mitochondrial DNA mutations. Okay, and in the interest of time, and because I want to show you a little bit more EM, I'll just say that we looked at the nucleoid machinery that's important for the replication of mitochondrial DNA and found, this is by Western blot and IP, that the nucleoid components were all modified with fumarate, this 2SC modification. This is the polymerase, the helicase, the polymerase subunit, the sort of histone. The only one that wasn't succinated was this SSBP1. It doesn't have any cysteines in it. We assay for the mitochondrial polymerase activity, and what we see is diminished processive enzyme activity for the polymerase and also diminished error-correcting activity for the mitochondrial polymerase. And so that leads us to conclude that what we saw in our cell lines, each of which has a different mutation, and interestingly, 2 of our cell lines are row 0 cell lines from these tumors. They have no mitochondrial DNA remaining, which was really befuddling us for a long time. I won't go into the details, but we see two phenotypes, a mutation phenotype in the mitochondrial DNA and a depletion phenotype. And we propose that this is because of this succination and alteration or inactivation of the replication fork in the polymerase that's solely responsible for the maintenance of mitochondrial DNA in the cell. So to summarize, we go from a functional mitochondrial DNA situation where respiratory chain complexes can assemble and be active, and they can oxidize glucose not only just to pyruvate but all the way to CO2 using the respiratory chain, to a phenotype in FH deficient tumors where we have loss and mutation of the mitochondrial DNA that precludes competent assembly of the respiratory chain and kind of burns in this phenotype of lactate fermentation and also anabolic metabolism by way of the penose phosphate pathway. So alternative uses of glucose that don't require respiration. The reason why this is interesting is looking further at these cell lines, when we look at respiration, this is a seahorse assay. Looking on the y-axis is respiration of our tumor cell lines, and the x-axis is time. And these 3 guys that are down here at the bottom aren't really respiring any detectable oxygen, whereas other tumor types, genetically defined tumor types, show some degree of respiration. So it really ties down the point that the respiratory chain is not working in our FH deficient tumors. So we wanted to ask this question: if our cells or tumor cells have broken respiratory chain and they're unable to extract oxygen from the bloodstream, is that something we can measure biophysically? And I'll just summarize with this slide. We used tools from Dr. Murali Charkuri. He built, and colleagues built, an instrument, electron paramagnetic resonance imaging, which can visualize free oxygen inside of tissues. And so when we look at 3 models of our FH deficient tumor, and the color here is oxygen mapping. So the more red, the more oxygen is in the tissue. And we compare to non-FH deficient tumor types. On average, we see a higher median PO2, a significantly higher tumor oxygen concentration. And when we look for hypoxic fractions, that's regions of the tumor that are below 10% millimeters of mercury, we see fewer hypoxic fractions in the FH deficient tumors. So I think this poses an interesting question. Maybe oxygen really isn't the rate limiting nutrient for growth of this tumor type, and maybe we can exploit that therapeutically, which I don't have time to discuss today. So the last minute or 2 that I'll spend is to talk about this kind of long-term project that Dr. Linehan has been interested in, as I mentioned, for many years, and Dr. Marino has been working with him for many years. Is these very difficult to deal with lesions that we sometimes see in our HLRCC patients, where it's either a very small blip in the kidney, not isoechoic, or it's a cyst, but a cyst in a normal patient, nobody worries about. The radiologist doesn't worry about. The cysts in our patients, we worry a lot about, because sometimes these cysts aren't just simple cysts, but they're actually cysts that are lined with neoplastic cells. So that's shown here in a very small lesion that we took out of a 20-year-old patient. And that you can see inside of that lesion, there are neoplastic cells that do not stain for the FH protein, but do stain for the 2SC protein. This was work done by Dr. Marino. So this little tiny blip that, some radiologists may just skip or say, "Oh, come back in a year, we'll have another look," can be very concerning in these patients. And here's another example of a patient that we had recently, a 28-year-old female who had several cysts in her right kidney. We, of course, got very worried about those cysts and performed a partial nephrectomy to remove them. Concerningly, here on the right-hand side, this is the 2SC stain, and it's mostly sort of connective capsule tissue. But you can see that there's regions that are staining positive for the 2SC, which means that the cells here have high fumarate. And so I think this is an example of a very early manifestation, really, because this is not a large, 3-centimeter tumor. This is a tiny microscopic region of a cyst. And recently with Ruqing and her colleagues, we've had this really awesome opportunity. And she just sent this particular image yesterday. But it has been the holy grail to get material from these cysts to do EM and other studies on, because Dr. Marino typically wants to take all of them, understandably, for pathology and diagnosis. But in this case, we were able to get a small piece of that cyst that I just showed you. And this is just an image of a tubular region of that cyst. And I think it's just fascinating to look and see more closely what do the mitochondria look like in here. So here's one cell from the cyst. You can see an enlarged mitochondria in here, another one here, some lipid droplet, and a funny looking nucleus. Here's another example of an enlarged mitochondrion. And there's a couple of them around in here. And then again, a more recent image that just came through was a particular cyst cell that was packed with mitochondria, but look at how the integrity of them is lost. So we have these big gaps inside of the mitochondria. We have poor cristae formation, but it doesn't quite look the same as the large solid lesions. So I think one thing will be interesting is just to see whether the cysts really do represent an earlier stage in pathogenesis. And so my last slide is really just to say about what we think happens in this disease. So if you think of the x-axis is kind of time. So under normal circumstances, we have low fumarate, Pol gamma is functioning fine, mitochondrial DNA is not mutating, the mutational burden of mitochondrial DNA is low, and the replication rate is normal, and the copy number of mitochondrial DNA is normal. This stochastic event that just happens where you lose the good copy of FH in the right cell at the right time sets off a cascade of events. And I think one of the earliest things is that fumarate rises, and that rising of fumarate causes things to become succinated. So polymerase gamma then becomes succinated. And once it's succinated and its function is altered, the mutational rate increases in the mitochondrial DNA. Much more slowly, I think the mutational burden increases because it takes time for these mutations to be introduced and to become homoplasmic or rise in heteroplasmy. At the same time, the replication rate decreases because of that lack of processive activity of the polymerase gamma. And so perhaps early, and we're going to be looking into this very, very soon, perhaps early on, these cells start to lose their mitochondrial DNA copy number. And that's something that we can measure using laser capture microdissected samples that we have in hand. So just to summarize, loss and mutation of mitochondrial DNA in FH-deficient tumors, deficiency in the respiratory chain, lactate fermentation. We found that oxygen levels are relatively high in this tumor type, which is interesting for many reasons. And something I didn't show you actually today was that how we could use a specific drug to exploit the high tumor oxygen levels in this disease. So Yuki Shibata did a lot of the in vivo work, and I'd like to thank Dr. Linehan, who's an outstanding colleague and a very astute.
Dan, I think
Developed
we lost
the EPR.
we lost you a little bit.
Oh, really?
Your audio, yeah.
Well, I was just thanking people, including yourself,
Okay. Thank you.
for all allowing these studies to go forward. And with that, I'll take any questions.